Good day, Sir Jeter. I am Marin Grace Clarang Acotanza, the first presenter of Group 6. For our final requirement in cell and molecular biology for teachers, our group will be discussing cell communication and signal transduction. In this presentation, we will introduce the basic concepts of how cells communicate with one another and how signals are transmitted to produce specific cellular responses. To make a multicellular organism, cells must communicate just as humans must communicate if they are to organize themselves into a complex society. And just as human communication involves more than the passage of noises from mouth to ear, so cell-to-cell communication involves more than the transmission of chemical signal across the space between one cell and another. And important note that communication between cells is mediated mainly by extracellular signal molecule. Before we proceed, allow me to read the objective first. At the end of a 60-minute discussion, the students are expected to achieve at least 80% mastery of the following objectives. First, define cell communication and signal transduction. Second, identify and describe the different types of intercellular signaling such as contact-dependent, paracrine, endocrine, and synaptic. And lastly, appreciate the importance of cell communication and signal transduction. So what is cell communication and signal transduction? Cell communication is how cells send and receive signals, allowing them to coordinate response to their environment. While signal transduction is the process that happens inside the cell after a signal is received, where the external signal is converted into a specific cellular response. Now let's continue with our discussion and general principles of cell communication. Long before multicellular organisms appeared on Earth, unicellular organisms had developed mechanisms for responding to physical and chemical changes in their environment. This ability to sense and react is the foundation of how cells communicate today. When we say unicellular organism, it refers to single-cell organism, while multicellular organisms are composed of many cells that specialize to form various tissues, organs, and organ systems. This diagram illustrates a simple intracellular signaling pathway activated by an extracellular signal molecule. An extracellular signal molecule binds to a receptor protein on the plasma membrane of a target cell. This binding triggers a cascade of reaction involving intracellular signaling proteins. These proteins activate effector proteins, which can be metabolic enzyme, gene regulatory proteins, and cytoskeletal protein. The activation of these proteins leads to a specific cellular response such as altered metabolism, altered gene expression, and altered cell shape or movement. Budding yeast cells responding to a mating factor. Similarly, yeast cells communicate with one another in preparation for mating. In the yeast Saccharomyces cerevisiae, a haploid cell releases a mating factor, which is a chemical signal that tells cells of the opposite mating type to stop dividing and get ready to mate. When two opposite cells join, they form a diploid cell, which can later produce new cells with different gene combination, helping the species to adapt and survive. This shows that even simple organisms like yeast use communication system similar to those humans, providing that cell signaling is a fundamental process of life. Extracellular signal molecules bind to specific receptors. This diagram shows the two main types of receptors and how they interact with different signaling molecules. Let's start with A, the cell surface receptors. As we can see on the slide, hydrophilic signal molecules, or those that love water, cannot cross the plasma membrane because it's made up of lipids. So instead, they bind to receptor proteins located on the cell surface. Once this happens, the receptor sends a signal inside the target cell. It's like a messenger na nagpapas ng information without entering the cell. Now for B, the intracellular receptors. From the word intra, meaning nasa loob or inside. These are for small hydrophobic molecules, meaning they can easily pass through the plasma membrane. However, since they don't dissolve well in water, they usually travel in the bloodstream attached to the carrier proteins. When they reach the target cells, they enter and bind to receptors found inside the cytosol or even the nucleus, directly affecting the gene expression. In short, cell surface receptors are for hydrophilic signals that stay outside, while intracellular receptors are for hydrophobic signals that go inside. There are 4 forms of intercellular signaling. First, we have contact-dependent signaling or membrane-to-membrane contact. This involves direct contact between a signaling cell and the target cell via a membrane-bound signal molecule. And second, we have paracrine signaling. A signaling cell releases a local mediator that acts on nearby the target cell or neighboring cells. Third, we have synaptic signaling. A neuron releases neurotransmitter into synapse to signal a target cell. And lastly, we have endocrine signaling. An endocrine cells releases hormones into the bloodstream which travel to distant target cells throughout the body. The contrast between endocrine cells and neuronal strategies for long-range signaling. Endocrine signaling. As we can see in Part A, endocrine cells release hormones into the bloodstream. These hormones travel long distance and only affect target cells that have the right receptors. So basically, this type of signaling is slower, but it can reach multiple target cells throughout the body. On the other hand, Part B shows synaptic signaling, which happens in neurons. Here, nerve cells release neurotransmitters across a synapse a very short distance to reach a specific target cell. This makes it faster and more precise than endocrine signaling because it directly targets one cell at a time. Extracellular signals can act slowly or rapidly to change the behavior of a target cell. Please rely on the diagram so you can follow my discussion. First, this is the extracellular signal molecule. It comes from outside the cell. Next, it will bind to a cell surface receptor protein. This receptor receives the signal and starts the communication inside the cell. Then, the intracellular signaling pathway carries that message throughout the cell. There are 2 ways the cell can respond, the fast response and the slow response. Fast response happens in seconds to minutes. It changes how existing proteins work, that's called altered protein function. As a result, the cell can change its movement, secretion, or metabolism. While the slow response takes minutes to hours, the signal goes to the nucleus and changes gene expression, DNA to RNA to new proteins, and that's what we call altered protein synthesis. Both fast and slow responses lead to altered cytoplasmic machinery, and finally altered cell behavior. For example, slow responses lead to cell growth and cell division. In summary, extracellular signals can act rapidly by changing existing proteins or slowly by making new ones. Both are helping the cell adjust its behavior based on what's happening around. Now let's talk about gap junctions, allow neighboring cells share signaling information. Okay, what is gap junctions? Gap junctions are narrow water-filled channels that directly connect the cytoplasm of adjacent epithelial cells, as well as of some other cell types. As we all know that epithelial cells, example of this is hair, skin, and nails. Here we can see two cells that are connected by what we call gap junction. A gap junction acts like a bridge or tunnel between two cells. As you can see in the middle of a gap junction, there are small openings. Through that, small molecules can pass directly from one cell to another. These small molecules include important signaling substances like cyclic AMP or CAMP and also calcium ions. These molecules help the cells communicate and coordinate their actions. So, when one cell receives an extracellular signal, that message can quickly spread to the neighboring cell through the gap junctions. This allows both cells to respond together in a coordinated way. An animal cell's depends on multiple extracellular signal molecules. Each cell type displays a set of receptors that enables it to respond to a corresponding set of signal molecules produced by other cells. These signal molecules work in combinations to regulate the behavior of the cell. As shown here, an individual cell often requires multiple signals to survive, as shown in blue arrows, and additional signals to grow and divide, as shown in red arrow, or differentiate, as shown in green arrows. If deprived of appropriate survival signals, a cell will undergo a form of cell suicide known as apoptosis or cell death. In simple terms, cells need constant communication with other cells to survive, grow, divide, or differentiate. Without these signals, the cell can't function properly and will eventually die. Different types of cells usually differently to the same extracellular signal molecules. When we say extracellular signal molecules, ito yung mga molecules na nasa labas ng cell na ginagamit para magpadala ng signal o message to the other cell. Even if the signal is the same like acetylcholine, which is shown in A, different cells respond differently depending on what receptors and intracellular machinery they have. So, here in heart muscle cell, there is a receptor protein na tumatanggap ng acetylcholine. Pag dumikit si acetylcholine sa receptor, nagiging signal ito sa loob ng cell na nag-decrease ng rate and force of contraction, meaning humihina at bumabagal ang tibok ng puso. In skeletal muscles naman, if acetylcholine bind here, it triggers muscle contraction, meaning skeletal muscles become active so they move or tighten. And lastly, salivary gland cell, there is also a receptor for acetylcholine. When it binds here, it triggers secretion or saliva releases, meaning gland cells are activated to release fluid like saliva. The fate of some developing cells depends on their position and morphogen gradients. The extracellular signal molecule in these cases during development is called morphogen. And in the simplest cases, it diffuses out from a localized cellular source, a signaling center, generating a signal concentration gradient. As shown in figure 15 to 10, cells adapt different fates depending on their position and morphogen gradient. But first of all, we need to understand what is morphogen. A morphogen comes from the Greek word morphe, which means form, and gen, which means generate. In other words, a morphogen is a signaling molecule that controls how cells develop into different types during growth. As we can see, cell A is closest to morphogen source, will receive highest concentration, and cell B is in the middle, will receive medium concentration, and for cell C, which is farthest, will receive lowest concentration. And when we say uncommitted cells naman, ito yung mga cells na wala pang specific function or identity turn into committed cells according to their position and gradient. The importance of rapid turnover. In graph A, when the synthesis of a molecule slows down, the concentration of that molecule also decreases. The red line shows a molecule that degrades quickly, it drops fast. The green line shows a stable molecule, it changes slowly. In graph B, when synthesis increases, molecules build up. Again, the short-lived molecule, which is the red line, reacts fast, while the long-lived one, which is the green line, responds slowly. This shows that fast turnover lets the cell adjust quickly to new signals. The numbers in blue on the right are the half-lives assumed for each of different molecules. The shorter the half-life, the faster the cell can change its response. The role of nitric oxide in smooth muscle relaxation in a blood vessel wall. In Part A, this is a simplified drawing of an autonomic nerve contacting a blood vessels. Inside the blood vessel, we can see the lumen. It is the hollow space where the blood flows. We also have endothelial cells, smooth muscle cells, basal lamina, and a nerve that sends signal. While in Part B, when the nerve is activated, it releases a chemical messenger called acetylcholine. Acetylcholine binds to our receptors on endothelial cells, which activates an enzyme called nitric oxide synthase or NOS. This enzyme converts the amino acid arginine into nitric oxide, also known as NO. Nitric oxide is a small gas molecule that quickly diffuses from the endothelial cells into the nearby smooth muscle cells. Inside the smooth muscle cells, nitric oxide binds to the enzyme called guanylate cyclase, which converts guanosine triphosphate or GTP into cyclic guanosine monophosphate or GMP. The cyclic GMP causes the smooth muscle cells to relax, leading to the widening of blood vessels, also known as vasodilation. As a result, blood can flow more easily, improving circulation. Here, we can see several examples of some non-gaseous signal molecules that bind to intracellular receptors. We have cortisol, a stress hormone that helps control metabolism and the immune response. Estradiol and testosterone, which are steroid sex hormones responsible for male and female characteristics. Vitamin D3, which is important for calcium absorption and bone health. Thyroxine, a thyroid hormone that controls metabolisms. Retinoic acid, which is derived from vitamin A and helps regulate cell growth and development. All of these hormones bind to receptors that belong to what we call the nuclear receptor family. These receptors act as transcription factors, meaning they can directly control which genes are turned on or off. Some members of this receptor family are called orphan nuclear receptors. These are receptors whose specific signaling molecules or ligands are still unknown. The nuclear receptor superfamily. Did you know that all nuclear receptors are proteins that can bind to DNA to control gene expression? That means, kapag na-activate sila, kaya nilang i-on or off ang specific genes sa loob ng cell. Okay, let's start with part A. It shows different examples of nuclear receptors like the cortisol receptor, estrogen receptor, progesterone receptor, vitamin D receptor, thyroid hormone receptor, and retinoic acid receptor. All of that receptors have similar structure. They have DNA binding domain, ligand binding domain, and transcription activating domain. Now, look at B. This is the inactivate receptor. In this form, the receptor is bound to inhibitory proteins, so it cannot activate genes yet. When a ligand or hormone binds to the receptor as shown in C, the receptor changes shape. The inhibitory proteins are released and co-activator proteins attach. This activates the receptor, allowing it to bind to DNA at a region called the receptor binding element. Once it bound, it can start the transcription of target genes, meaning it turns on specific genes to produce proteins. In part D, we see the actual structure of the receptor. When the ligand binds, the receptor closes around it, holding it tightly parang may lock and key mechanism. In short, nuclear receptors like switches inside the cell. Kapag walang ligand, inactivate sila. But if there's a hormone that binds to a receptor, they activate genes and control many important cells functions like metabolism, growth, and development. Figure 15 to 15, an example of primary and secondary responses induced by the activation of a nuclear hormone receptor. Let's start with part A, primary or early response. When a steroid hormone enters the cell, it binds to its receptor to form a receptor-steroid hormone complex. This complex then attaches to the DNA and activates primary response genes. As a result, the cell begins to produce primary response proteins. These are the first proteins made after the hormone signal. In part B, we have the secondary or delayed response. Some of the primary response proteins can turn off the primary genes, while others turn on a new set of genes called secondary response genes. These new genes produce secondary response proteins, which create longer-lasting effects such as cell growth or differentiation. There are three largest classes of cell surface receptor proteins. First, we have ion channel coupled receptor. Second is G protein coupled receptors. And third, enzyme coupled receptors. Later on, I'll discuss it one by one. Let's begin with ion channel coupled receptors, also known as transmitter-gated ion channels or ionotropic receptors, are involved in rapid synaptic signaling between nerve cells and other electrically excitable target cells such as nerve and muscle cells. A signal molecule binds to the receptor, causing a channel to open and allowing ions to pass through. G-protein-coupled receptors act by indirectly regulating the activity of a separate plasma membrane-bound target protein, which is generally either an enzyme or an ion channel. And enzyme-coupled receptors either function directly as enzymes or associate directly with enzymes that they activate. They are usually single-pass transmembrane proteins that have their ligand binding site outside the cell and their catalytic or enzyme binding site inside. A hypothetical intracellular signal pathway from a cell surface receptor to the nucleus. As we can see in the figure, cell communications start when an extracellular signal molecule binds to a receptor protein on the cell surface. This binding activates a series of intracellular signaling proteins inside the cytoplasm, and that's what we call a signaling pathway. Now, let's go through each process shown in the A signal from the cell surface receptor is passed through a series of signaling proteins inside the cell. These signals are transduced, amplified, and modulated as they travel toward the nucleus. The process results in changes in gene expression, turning specific genes on or off. The final effect depends on multiple external and internal signals affecting the cell. Effector proteins, such as regulatory proteins, control how the cell behaves. Some proteins, like scaffold proteins, can perform multiple roles, helping organize and anchor signaling molecules. Effector protein activation to gene transcription. This is the final response. The signal reaches the nucleus and activates genes to make the proper cell response. There are two types of intracellular signaling proteins that can act as molecular switches. First is the signaling by phosphorylation. A protein kinase covalently adds phosphate from ATP or adenosine triphosphate to the signaling protein, and a protein phosphatase removes the phosphate. Although not shown, some signaling proteins are activated by dephosphorylation rather than by phosphorylation. When you say dephosphorylation, it is the process of removing a phosphate group or PO4 from a protein or molecule. It is done by an enzyme called protein phosphatase. Yung phosphorylations naman is kabalagtaran lang sa dephosphorylation. Second is the signaling by GTP binding. In this case, the protein is activated when GTP or guanosine triphosphate binds to it and becomes on, and it turns off when GTP is hydrolyzed to GDP or guanosine diphosphate, meaning water helps break one phosphate group off from GTP, turning it into GDP and inactivating the protein. The regulation of monomeric GTPase or guanosine triphosphatase. The monomeric GTPase is inactive when it's bound to a molecule called GDP. To turn the switch on, we need a guanine nucleotide exchange factor or GEF. The GEF activates the protein by stimulating it to release its bound GDP. Once the GDP is released, the protein is free to bind a new molecule GTP. The concentration of GTP in the cell cytosol is about 10 times greater than concentration of GDP. So, the protein rapidly binds to GTP, then the protein becomes active. Now that the protein is active, as what we can see in the active monomeric GTPase, it can perform its cellular function. But what turns it off again? That's where the GTPase activating protein or GAP comes in. GAPs inactivate the protein by stimulating it to hydrolyze its bound GTP back into GDP, which remains tightly bound to the inactivate. Now let's proceed to signal integration. Based on the image, we can see the extracellular signals A and B activate different intracellular signaling pathways, each of which leads to the phosphorylation of protein Y but at different site on the protein. When we say phosphorylation, it is a biochemical process in which a phosphate group is added to a molecule. Protein Y is activated only when both of these sites are phosphorylated, and therefore it becomes active only when signals A and B are simultaneously present. Such proteins are often called coincidence detectors. We are done discussing the signal integration. Now let's delve into the three types of intracellular signaling complexes. First, the performed signal complex on a scaffold protein. Here, a large scaffold protein holds a receptor and inactivates signaling proteins together. When a signal molecule binds, the receptor is activated and the pre-assembled complex is ready to transmit the downstream signal. Second, the assembly of signaling complex on an activated receptor. This mechanism is a bit more dynamic. Why? Because it's like on-demand assembly. Initially, yung inactive receptor is naked. So to speak, walang naka-attach na signaling proteins. The magic happens only after the signal molecule binds. Here, the activated receptor phosphorylates itself at multiple sites, which then act as docking sites for intracellular signaling proteins. Finally, For Part C, the assembly of signaling complex on phosphoinositide docking sites. Here, as we can see in the image, the signaling complex hindi lang sa receptor nag-a-assemble but directly sa inner part ng plasma membrane. When activated, the receptor causes specific membrane lipids, the phosphoinositides, to become hyperphosphorylated, which then serve as docking sites for specific intracellular signaling proteins which can now interact with each other. If you wondered, what is phosphoinositides? Here are the simple explanation. Phosphoinositides, or PEPs, are crucial cell membrane lipids phosphorylated forms of phosphatidylinositol, acting as vital signaling molecules that regulate nearly every aspect of cell life, including growth, membrane trafficking like Bessel's movement, cytoskeletal control, and communication, by recruiting specific proteins to different membrane locations to direct cellular process. They're like cellular zip codes, telling proteins where to go and when to act. And that is phosphoinositides. Now let's talk about intracellular signaling networks usually used of feedback loops. There are two types of feedback loops, the positive and the negative feedback loop. A positive feedback loop in a signaling pathway can transform the behavior of the responding cell. If the positive feedback is of only moderate strength, its effect will be simply to steepen and increase the response to the signal. In contrast to negative feedback loop, it counteracts the effect of stimulus and thereby abbreviates and limits the level of the response, making the system less sensitive to perturbations as with positive feedback. However, qualitatively different phenomena can be obtained when the feedback operates more powerfully. Feedback loop is a process in which the output of the process acts back to regulate that same process. In these simple examples, a stimulus activates protein A, which in turn activates protein B. Protein B then acts back to either increase or decrease the activity of A. In positive feedback, the output stimulates its own production, while in negative feedback, the output inhibits its own production. Its goal is to make our body stable or achieve homeostasis. Homeostasis is the process of maintaining balance. Here, the positive feedback mechanism is switching the active enzyme to a more active enzyme. We can see in the image that the enzyme is still inactive because there is no signaling ligand yet. Ligand means signal molecule that binds to a specific site on a protein, like an enzyme or receptor. When the ligand arrives, it will attach to the enzyme and it will become active. The active enzyme will now convert the substrate into product. As the product increases, two of these molecules return and attach to the enzyme. This is the positive feedback, because the product activates the enzyme even more. Because of this, it becomes a very active enzyme, so production suddenly increases like a switch that suddenly turns on. And that is a positive feedback mechanism giving switch-like behavior. Let's proceed to some effects of simple feedback. In the first part, we see a positive feedback loop. When a signal kinase activates enzyme kinase, it acts back to promote its own phosphorylation and activation. The basal activity of the I-phosphatase, the phosphorylase, activated E at a steady low rate. In part B, the top graph shows that without feedback, the activity of E kinase is simply proportional with a short lag to the level of stimulation by the S kinase. Control the feedback. It is response only when signal is present. The bottom graph shows that with a positive feedback loop, the system is bistable, that it is capable of existing in either of two steady states. The transient stimulation by S kinase switches the system from an off state to an on state, which then persists after the stimulus has been removed. In negative feedback naman, the activated enzyme kinase phosphorylates and activates the I-phosphatase, thereby increasing the rate at which the phosphatase dephosphorylated and inactivates the phosphorylated E kinase. So this serves as a control mechanism so that the response is not excessive. In graph D, pinapakita nito na kapag short delay, mabilis bumaba ang activity. Ibig sabihin, mabilis ang pag-regulate. Pero kapag long delay, nagkakaroon ng oscillations, parang on and off cycles habang may signal pa. For the final part of my report, I'll be explaining the different ways in which target cells become desensitized or adapted to an extracellular signal molecule. First, we have receptor sequestration. Sequestration means isolate or separate. In this process, the receptors are temporarily removed from the cell surface and placed inside endosomes. This prevents overstimulation while allowing the cell to reuse the receptor later. Second, receptor downregulation. Unlike sequestration, here the receptors are destroyed in lysosomes. This permanently decreases the number of receptors available for signaling. Third, receptor inactivation. Here, the receptors remain on the cell surface, but they are chemically modified, such as by phosphorylation or methylation, making them inactivate even if signal molecule is present. Fourth, inactivation of signaling protein. This occurs inside the cell, where the signaling proteins are deactivated, stopping the transmission of the signal. And lastly, inactivation of inhibitory protein. This is where the cell produces an inhibitor that blocks the signal transduction pathway and preventing further response. In summary, these mechanisms are essential for maintaining cellular balance and ensuring that cells do not overreact to continuous stimulation. I guess that's the end of my report. Thank you so much for listening and I hope you learned something. Again, this is Mern Grace Clarang Acotanza.